Hello, everyone. Welcome to BioConference Live. Just as an introductory comment, let me note that at the end of this, about 40 minutes from now, we'll have a question and answer period. So start saving up your questions and answers right now. In fact, you can enter them online in the Q&A box if you're not familiar with that. Familiarize yourself with it now. So the topic for this BioConference Live talk is technology and open access to cohorts for combining genomes, other omics, environmental, and trait data. This is my conflict of interest slide. I have many organizations that have helped bring the technologies that we've been working on into the public domains for the public and for researchers. So the next slide is about this revolution in genomics and omics more broadly defined here. We're talking about epigenomics, proteomics, transcriptomics, and so forth. And this is not just in analytic, but it's also synthetic. It affects our ability to manipulate the omes. And we're going to talk about one particular open access route, which is the Personal Genome Project, or PGP, and how this impacts our transition to clinical quality transfer and therapeutic prioritization. And then I'll give you two examples of new technology, including in situ sequencing. On the third slide here, we have something that's often confused in this revolution of, especially on interpreting inherited genomics. There's also pharmacogenomics and cancer genomics, but specific on inherited genomics. There are now over 2,400 genetic diseases which are considered highly predictive and medically actionable, so much so that this is the practice of medical genetics and is now suitable for high-throughput sequencing methods. In the past, these have been restricted to people with family histories and one gene at a time, largely for economic reasons and background information. We now are getting that background information and we can deploy it. These affect at least 10% of us, and they are, as I said, highly predictive and medically actionable. And this has been going on since the mid-90s. Here's an example of one of the Personal Genome Project participants. We'll a little bit more of that project in a moment. This is John Lauerman, who described this in his Bloomberg column. He had a JAK2 mutation, which is not in his inherited genome, but was developing somatically in his blood and could lead to one of several different diseases. And this was caught far earlier than it would have normally been caught because we were sequenced his blood genome in this project. Steven Pinker has also commented on this in the New York Times and other places. His experience was finding out potential risk factor for hypertrophic cardiomyopathy, which is a disease that strikes healthy individuals without warning, sometimes resulting in very premature death, even among athletes. Now, this revolution, in principle, is invading do-it-yourself biology, not just the research market, not just direct-to-consumer, but even kids. Here is a do-it-yourself kit for your kids, and there are many other ones that are aimed at all ages, where your kid can figure out who the father is or other innocent activities. This photo from the Wall Street Journal is a student who spent 3 summers in our lab, Ann West, who analyzed all 4 of her family members, including her father, who had factor V Leiden, which is a risk factor for long plane trips, which he does quite a few of. But more significantly, we have a growing number of successes of new personal genomics, not just the conventional medical genetics that might require up to $3,000 per gene test. Now we can get entire genomes for $3,000 to $4,000. And we have examples like Nick Volker, who had misdiagnosis of all kinds of intestinal surgeries up until one of his pediatricians decided to get a whole genome or exome sequence done. And then it changed to a focus on the immune system, and a blood stem cell transplant resulted in a huge change in his prognosis and more or less normal life. The Beery twins were misdiagnosed with cerebral palsy, and then slowly moved towards whole genome, whole exome sequencing to find that all that was needed was a dietary change to dopamine and serotonin analogs. And finally, I should note that the Food and Drug Administration moves quite promptly when we have technologies that actually work. For example, here's a very rare cystic fibrosis allele that for which there is now a drug that's specific for that particular DNA sequence, and the FDA approved that in a 3-month period, and they're quite rightfully proud of this achievement at the Food and Drug Administration. I also want to note PGED is a force in the community now to get this education deployed into high schools across the United States and the world. So where do we go from here? We are not merely talking about going from your inherited genome to your traits. We're not talking about perfect prediction in the absence of all other medical diagnostics. It's more of an integrated community, and we can, so we do have, and it's now 1 million times less expensive to do the whole human genome, and so it's now affordable to many people around the world. But we have the opportunity to fill in additional data, classical medical traits and measurements, as well as a variety of new ones that are becoming wireless 724 monitoring and so on. But the omics that made genomes 1 million times cheaper also makes many environmental and metabolic measurements internal to your body also 1 million times less expensive. And we'll see those prices slowly drop in the marketplace. And this includes the microbiomes that surround you in the world, your immune response to them, the metabolic response of your body to these inflammatory events, foods, allergens in your environment, and so forth. All of these have omic components. And how we respond developmentally in terms of aging, these are reflected in the epigenomics, meaning the RNA and protein in the various tissues of your body as you develop and age. Finally, imaging is something that we'll talk about, where there's a merger now of various imaging methods and the omics methods. So this project, there is only one project that I'm aware of worldwide that is capable of sharing data broadly. This open access project is, you can find information about it or volunteer at personalgenomes.org. It has some international components, and it is distinguished by individuals who've been consented that are willing to have their genomes, their cells of various primary and cell lines available through worldwide without restriction. They're available for commercial use without any strings attached, and we have approval for 100,000 volunteers in the Boston branch alone, and then there are many other branches. And the main requirement is that people get 100% on an entrance exam, which is considerably easier now than it used to be, and is just to make sure that you've read the consent form, essentially. Now, what we're talking about here is measurements, not merely the omics measurements, but an increasing number of wireless measurements that measure how the omics plays out through and how you differ from day to day and second to second. And these include measurements of your EEG, of brain activity and sleep activity. Some people fall asleep during the daytime. fMRI is another brain measurement, typically not done wirelessly. Cardiovascular measurements, ultrasound, and skin monitors. These are all making giant leaps forward, and many of them will become increasingly considered an extension of your cell phone. Going back to genomics and the omics measurements, we are looking for exceptional individuals where each of us have rare alleles that are specific to ourselves or our families, and some of these are not just deleterious, but they're rare protective alleles. You might associate these with people who live for over 110 years, for example. And here's one that has been reproduced, kind of a standard for scientific practice, to be reproduced in animal models. So the humans that exhibit this, so this case Flex Willer, does also work out, but you can see the lean body mass, which is somewhat independent of diet, is reflected in the cow, the dog, and the mouse models for this. So this double null of the myostatin gene has these very interesting phenotypes of decreased body fat and decreased atherosclerosis. So this is an interesting genotype to explore, and there are many more. So when we're looking beyond correlations to causative alleles, and in particular these highly protective rare alleles, which are not easily captured in most of the standard Personal Genome Project methodologies. And so other examples beyond the myostatin double null I just talked about, where there are LRP5 gene alleles, which can be in the heterozygous state, meaning both mutant and wild type from mother and father, which result in extra strong bones capable of breaking surgical drill bits. PCSK9 gene alleles are extreme significance and importance as we get 85% or so lowering of coronary artery disease risk. A huge risk change. CCR5 we'll talk about in just a moment, but it and FUT2 are examples of alleles that relatively rare, not the major allele in the population, that make the individuals that have double nulls resistant to a variety of viruses, such as norovirus in the case of FUT2 and HIV in the case of CCR5. So I'd note this blog down at the bottom that gives more background information at personalgenomes.org. So drilling down a little bit deeper on the CCR5 example, we have double null has been observed to cause resistance to a large class of HIV, AIDS-causing viruses, and this observation in natural populations, they're almost non-existent in Asian African populations. But the few individuals that have this prompted now a phase 2 clinical trial from Sangamo making a zinc finger nuclease, a designed protein enzyme that will cleave specifically in the CCR5 gene in cells derived from the body of these patients, blood stem cells that can be then manipulated genetically precisely. This is not the old-fashioned gene therapy where viruses would integrate randomly throughout the genome, but these are very precise change in just one gene, both copies from maternal and paternal. So that now we have both copies of your CCR5 HIV co-receptor are gone, and so even a patient that started out with full-blown AIDS, possibly even drug-resistant AIDS, could now become fully resistant because the few T-cell precursors in that autologous transplant are now fully AIDS-resistant, and the ones that were not affected slowly die off, and we have people that survived not this clinical trial, but previous stem cell transplants for over 5 years. Now, another way to impact our interaction between our immune system and the microbiome is we can measure both directly the microorganisms, which has advantage for drug resistance. Drug resistance is not easily observed in the alternative set of diagnostics, which focus on our immune response. We use diagnostics that are focused on very specific pathogens like HIV or tuberculosis, TB, by looking at the body's immune response to them. That's appropriate if you have a hit and run or a very rare, hard to detect virus or bacterium. But you have these two options of doing diagnosis at either the microbial level or the immune. Now, we'd like to generalize the immune tests, just as we've generalized the inherited genome tests from single gene tests where you have pre-existing conditions to whole genome tests. Now we want to extend single microorganism tests to your entire microbiome, or at least a very large set of potential pathogens. Now we do this more general version of it by sequencing your entire immune response from a peripheral blood test. Depending on whether we're looking for the cellular response or antibody response, we can do this somewhere in the range of 1 microliter to a milliliter of blood. This diagram is kind of a textbook diagram of the human immune genes in their germline and their rearranged state. We have now on the order of 24 of the heavy chains rearranging with the 23 D regions and 6 J regions. So this VDJ rearrangement combined with a light chain can produce about 3 million combinations. But in addition, as you connect these, you fill in with novel nucleotides, which can range from 0 fill-in to 140 some base pair fill-in via de novo template-independent polymerase action of the terminal transferase polymerase. And so the incredible diversity that's generated here, not just by genomic rearrangements of existing gene fragments, but these new components that sometimes called N regions produced by terminal transferase. And we can monitor these. We've done this in collaboration with Roche, and this was work from Yuri Laserson and Francois Vignon, where we can do this on blood samples as a function of time and keeping track of specific events such as the vaccines or exposure to diseases. So this falls into in this omics revolution orientation slide into the epigenomics and proteomics and immunomics. So I mentioned that these N regions where you get rearrangement of the V and the D and the J can be quite large. And here's an example of one of those large ones. It's 84 base pairs long. It can be as little as 0. This particular one had provided a second way, in addition to the one we talked about earlier. This is a way of getting neutralizing antibodies that makes rare individuals in our population resistant to HIV. And these neutralizing antibodies have been characterized and are the basis of a new way of obtaining immunity, not via vaccines, where you count on the vaccines producing reproducibly in a large fraction of the population an immune response, which has been very difficult in the case of HIV, to a new procedure where instead of posing the problem to your body, you give it the answer. You can say, instead of having the body try to figure out what's the right antibody to make to deal with HIV or HIV vaccine, you give it the answer, which is this is an antibody that's worked, in 1 in a million individual. Now we can deploy it along a broader set. David Baltimore and his group and other groups are excited about this pathway and are testing it by injecting AAV vector loads with these antibody genes into muscle cells. Now the muscle cells do something that's very unnatural for muscles. They imitate B cells and produce antibodies. But this looks very promising in the rodent models so far, and we look forward to primate and human testing. Now that rare antibody is rare in part because it was so long. That long usually would be a deep groove, and now it's a long protrusion from the antibody, can snake into a conserved region of the HIV surface glycoprotein, and since it's conserved, it's one of the few things that the HIV virus, which is highly mutable, doesn't change. But that required this, not necessarily something that every person body would come up with in response to a vaccine. It required in this distribution. This particular distribution is the length of these CDR3s, these position where the V and D and J come together. This is a part of a time series analysis for vaccination trials. This is from the Personal Genome Project not an HIV project. And you can see factors of 10 along the y-axis and lengths ranging from 0 to 145 base pairs in this highly variable region. And you can see that 84 is well into the region where these antibodies tend to be unique for a particular individual. And we detect these in a variety of samples that we're doing with this immune sequencing, which is now only feasible because of the great drop in the cost of genomics. So we've been following a vaccination time course for some of these individuals for over 3.5 years now. And you can see here's a little piece of that time series where we've aligned 3 different individuals at a time 0 of a seasonal flu vaccine. And you can see for the 2 younger ones at the bottom a ballooning out, sort of a textbook expansion of the flu responsive antibodies at around 7 days after vaccination, and then the top one, which happens to be my immunome, for some reason or other has a much less distinctive one. The color coding here is indicates that we can follow individual antibody gene sequences no matter where they go in the body, no matter how long the time that elapses, and even if other parts of the immune gene, antibody encoding gene changes with time, we can still monitor this sort of natural barcode tag, which is this hypervariable region, throughout time, and color code anything that's seen in multiple time points. Now, we'd like to combine that deeper understanding we have of the heavy and light chain. We can couple now those two together, and we can now couple them to specific antigens. So we can take either a natural fluid containing antigen binding molecules like antibodies, or we can take these hybrid molecules that we rescue from peripheral blood and manipulate to nucleic acid at the cDNA level. Anyway, we could take any library of binding molecules, let's call them antibodies, and a library that we make of potential antigen molecules. They can be antigens from your own body. These would be autoantigens, self proteins to which you should not be responding immunologically. And we can make a library of these antigens and bind them to a library of antibodies or other binding molecules and pull down the specific binding events, and then correlate which antibodies bind to which antigens. And this is done in collaboration with Ben Larman and Steve Elledge in the Harvard Department of Genetics. And this can be expanded, and is being expanded now, from human auto-antigen reactivity to bacterial, viral, and allergens and foods, which may or may not be allergens. So you can make these large libraries of potential antibody binding that can bind these antigens and could potentially cross-react. So this is, I think, a very interesting new technology, and again, benefits greatly from our progress in omics. So how, kind of put the cart before the horse intentionally here, wanted to motivate, how did we get to this point where we take for granted all these new omics technologies and can use them in various novel ways? Well, we have a variety of new technologies. 2 that I'd just like to mention here is this in situ sequencing and nanopore sequencing. How are these getting closer to the clinic, where we have to have very high accuracy? When we make predictions about how quickly technologies go, we can err either on the optimistic or pessimistic side. When we were extrapolating from about the time that we were finishing off a very rough draft of around the year 2000, a rough draft of the human genome, if we took the stance of a very aggressive exponential curve, such as the exponential you get in the electronics and computer industry of 1.5-fold multiplicative every year, that seemed like a pretty aggressive curve, and that would result in an affordable genome in about 40 years from that $3 billion genome success. As it turns out, that curve was, even though it seemed quite aggressive, was actually on the pessimistic side. Instead of being 40 years, it was more like 6 to 10 years. And there's a change in slope. These are factors of 10 in base pairs per dollar on the y-axis and years on the horizontal axis. And so what we're seeing is prices dropping from around $2,000 a year to then the hundreds of dollars per human genome in the next year or two. And there's really no let up in this. Eventually, there will be a plateau of some sort or other, but it's quite interesting to follow this and its impact on a variety of other types of omics. The reason this happened is we moved from a monopoly, sort of a complacent single technology, to now dozens of different technologies represented both academically and in this case 44 commercially supported technologies over the past years. Not all of these are in existence at the same time. In the red box, the ones that are 9 that are currently available either as a service or as an instrument, and in green are ones that will hopefully be available within the next year or so that are focused on nanopore technology. The one in the red box tend to be focused on either multi-molecule or fluorescent technologies, sequencing by synthesis, they're sometimes called. Now, when we compare these two methodologies, we keep in mind the progress that's already been made. We've gone from this sort of centimeter scale sequencing technology on gel electrophoresis, where I did my graduate work with Wally Gilbert, to now hundreds of nanometer dots in grids, which can be detected fluorescently, getting close to the conventional fluorescent microscopy limit. This has been done we've had a number of papers in collaboration with Roddy Dramanik at Complete Genomics on this, including one recently in Nature where a error rate of 1 in 10 million was established. But looking forward, we have this nanopore technology as a possibility for getting exceptionally long reads, and we just need to watch and work on the technology to improve the quality, which I think, as far as I can tell, is as improvable as any other. That we have now handheld size prototypes of these. They're close to disposable in the order of hundreds of dollars, and some of the technology that previsions this back in the 90s of using polymerase in concert with a pore to measure the ion conductance as potassium or sodium ions go through the pore, and double-stranded DNA can be either inserted directly, stripped, so the single strand is going through the pore and interfering with the ion conductance, or you can have small molecules released from the single or double-stranded DNA going into the pore as another way of measuring it. A number of academic groups have contributed to this, including Jans Gundlach's group and Hagen Bailey's, and Oxford Nanopore and Genia are two of the commercial ones. And demonstrations have been announced, although there's nothing in the scientific literature yet, of up to 100,000 base pair reads. This is extremely important for filling in long gaps, and we expect to see the error rate improve from currently somewhere around 4% and to much higher qualities. Nevertheless, there are some things that are not immediately accessible to this new nanopore technology, and I'll give an example of that, where it looks like the fluorescent methodologies that are already in play may be immediately applicable. And we call this fluorescent in situ sequencing, or FISSEQ, and I'll give some examples of that in just a moment. Now, part of the motivation for in situ sequencing was being able to get very long continuity, not over hundreds of kilobases, not over hundreds of bases or 100 kilobases, but over hundreds of megabases, basically over the whole chromosome. There are 3 ways of doing this. The 2 that are in the most recent literature are using plasmids or bacterial artificial chromosomes or sheared DNA, where one can work from as little as 10 cells or even 5 cells as your input source. And this is not only feasible, but it's desirable to do a very small number of cells where you distribute the DNA over, say, a 384 well multi-well plate, and about 0.1 genome equivalent, about a tenth of a genome equivalent per well, whole genome amplification, and then barcoding and going into standard complete genomic sequencing, or for probably other methods as well. And this results in a long haplotypes on the order of megabase size haplotypes, and very high error rates, 1 in 10 million. So that's recently published in Nature. Extending that from the now millions of bases, a couple million bases, to whole chromosomes, it was demonstrated using this in situ sequencing methodology, using metaphase chromosomes, where you have the entire chromosome separating the maternal and the paternal, or separating one chromosome from many others. And you can ask a series of sequencing questions, essentially a series of sequences from each chromosome as if it's a dot. And this allows us to get haplotyping from telomere to telomere, and validated in this slide using a trio, which is yet another way of getting haplotypes, but unfortunately one that's not always available. But to get it from an individual where you may not have family genetic resources, you can do this dilution method of metaphase chromosomes. All right. And just a reminder, as we in the middle here, is keep your questions, start entering them online. We'll be there very soon. So finally, this in situ sequencing, extended now from chromosomes to other aspects of human biology. Here is where we're doing individual cells. We're looking for cancer cells that are not only different from the normal cells in your blood or in tissues by virtue of being cancerous and proliferative, but they're also different that they are now drug resistant. This is from a study that we did in collaboration with George Daly, where we're looking at the BCR-ABL tyrosine kinase change in these cells that makes oncogenic transformation. And this kinase is initially sensitive to the drugs and becomes resistant. So the little green dots in here you can see in some of these images are cancer cells that are drug resistant. So we're detecting a one base pair difference that makes both the difference in the drug resistance and allows us to do the diagnostic where we can detect a very rare cell. And the objective here is to detect those cells as early as possible in the parts per million and hopefully parts per billion in blood or tissues. So that's one other application of chromosomes, haplotyping, finding rare cancer cells or rare drug-resistant cancer cells. The final application is in characterizing your epigenome, how your body is responding to development and aging, and what the potentialities of your personal genome is to the responsiveness of your tissue to drugs and so forth. And to do this, we want to get access to all of your cells, but this is not feasible for living individuals, patients. So what we do in the Personal Genome Project is we establish biobanks of particularly useful cells. Either we have primary tissues available for saliva, blood, skin, and so forth, and then we have derived cells, particularly useful for these induced pluripotent stem cells, which have allow us to get access to a wide variety of cell types. Here's some data from the quality control QC from the Coriell Biobank, where 9 different tissues are routinely assayed by pathological histological examination. Here is showing the 3 major cell lineages: endoderm, ectoderm, and mesoderm. And you can see these neuronal rosettes, pigmented cells, ductal cells, and so forth, beautifully organized within these teratomas that are experimentally induced within mice. We can also take these human cell lines and make more complicated organized tissues in vitro For example, the personalized organs on chip. The organs on chip project is spearheaded by Don Ingber and Geraldine Hamilton at the Wyss Institute of Biologically Inspired Engineering, where I'm a co-worker. And together we're trying to combine the organs on chip with the Personal Genome Project to make these personalized organs on chip, which will be of value to drug testing. These organs on chips have multicellular components. They have mechanical components where you can actually have multiple phases. For example, in the lung, you have an air passage, lung epithelial layer, alveolar, so on. Another layer, and then endothelial, and then blood. And the mechanical forces that come along with breathing are simulated. And without these mechanical forces and correct geometry and extracellular matrix and so forth, you don't get proper cellular differentiation and physiological behavior. So this is a very exciting trend, and I urge you all to tune into it. Now we can analyze the faithfulness of those synthetic laboratory organs on chip with natural cells in as deep a manner as we wish. And in part, this is because this in situ sequencing method that I showed earlier for chromosomes and cancer cells can also be applied to the RNA for multiple RNAs at once. We have not yet achieved sequencing on every RNA of every cell in a specimen, but clearly it's capable of the kind of next generation sequencing that we've been talking about, which is in turn capable of doing RNA sequencing, getting multiple splice forms for essentially all the RNAs, even microRNAs present in a cellular sample. This is now not only single cell, where you might dissect the cell off of a slide, make a library, put it back on another flow cell slide for sequencing. Now we leave it in place on the slide and sequence it where each dot, which was a single molecule, is left in place, so you get the geometric context and the relationship to other cells. You get subcellular resolution, and many RNAs are unevenly distributed in physiologically meaningful ways throughout the cell, and this is a new technology that will allow us to get it. Now, what's limited in situ in the past is the limited number of colors, typically 1 or 2 or 3, maybe even 4. But with in situ sequencing, you now get to reuse those 4 colors over and over, just as you do in next generation fluorescent sequencing in general. And so, in effect, you get not 4 colors, but 4 to the n colors, where n is the number of cycles of sequencing. So we've now done this for 60 cycles, so you can think of that as 4 to the 60th power, or you can think of it as a barcode that's unique enough that you can tell which dot corresponds to which messenger RNA. And here we've taken a challenging test, we being Mats Nilsson and Jay Lee, Joyce Yang, and Rich Terry and our team. Challenging test where we have 2 cells that are very similar to one another. These 2 fibroblasts differ by 1 base pair, and the difference between the green and the red dots, each of them represent a single RNA molecule. You can see that delimited by the cell boundaries, that one cell will tend to have only red and the other one only green, which means they differ by 1 base pair in 1 messenger RNA type, and you can see every instance or many instances of single messenger RNA molecules in these 2 different cell types side by side. But that's just using 2 colors. We have, as I said, effectively 4 to the 60th colors available here. These can be lightly amplified so that they still are dots as far as the resolution of the microscope is concerned. The light amplification, however, makes them very strong signals that are easy to detect for sequencing. These can be immobilized in a variety of novel fixatives that we use for this, and this cross-linking allows us to have the cDNA covalently attached to the plastic-like protein cross-link matrix so that we can get these many rounds of sequencing by synthesis in a fluorescent mode, either sequenced by ligation or by polymerase. Here you see some three-dimensional we've done confocal and other microscopic measurements here. And we have this is showing how over 60 cycles we get essentially no change in signal to noise. We reproducibility of this is quite high, and this gives us the multiple colors that we need for unambiguous identification of each messenger RNA. Now, if we have information overload, there are two solutions to this. One is improving the resolution of the microscopy, and the other is doing selective sequencing of subsets of the RNAs. By doing subsets, you can do multiple subsets and eventually get the full set. But at any given moment, you're only looking at a subset, and so the viewing image plane is less complicated. Also, the thinner the sections, if you're doing sectioning, the thinner the sections and the less complicated is the image. And you can see at the very bottom of the slide how as we get more and more specific subsets, we get a smaller and smaller number of these detected RNAs with 4 colors at a time. So that's one approach, is by doing molecular tricks to select subsets by using semi-random primers that select or specific gene-specific primers to select subsets, ultimately getting as close to full transcriptome as you would like. And then the second method is using super resolution. This can be achieved either by optics, which we've done in collaboration with Lightspeed Genomics, where they have a ring of spatially patterned light provided by laser patterning, and you can see this huge improvement from the raw images in the middle to the aperture optics on the far right-hand side of the slide, where you get these sharp dots, maybe tenfold improvement in resolution here due to optics. And finally, so that's one method. The other method is using flickering fluorescent molecules, such as the Nikon Storm that our colleague at Harvard, Xiaowei Zhang, has developed. And this has somewhere between 10 and 20 nanometer resolution, which is really quite extraordinary for these in situ visualizations, and hopefully will be quite useful for in situ sequencing. So just in conclusion, I would just use this slide as a convenient way of encapsulating and summarizing where we talked about it. We'll also use it in the question. It'll stay up during the question answering session. Hopefully you've been writing in your questions already online. Where we've been on this little journey today is looking at this special cohort at personalgenomes.org. We urge you all to consider volunteering for this, where we can capture human genomes, environments, and traits and share them broadly without restriction, without restriction of having to do with re-identification or commercial uses. We not only have their personal inherited genome, but we have growing resources on their microbiome from 5 different body sites, the immunological response to their environment, including food and other metabolic components that can influence immune response and inflammation, and then how this microbiome and immunome affects their developmental and aging process in the epigenomes, RNA and protein and immune components. And finally, this imaging, not just the in situ sequencing I was talking about, but very complicated physiological monitoring, such as functional magnetic resonance imaging. And this we consider part of the traits, and the trait data set for these individuals is growing. And as more and more is learned about these individuals, they become like Rosetta Stones. They become more valuable and yet attract even more attention. So I'm going to leave this slide up as I open it up for questions and answers. Please feel free to jump in at any time, and I'll just start with the ones that we have already here. So we have one from Karina Nikoloff at Affymetrix, which asks, how can you detect single base differences in RNA? How long is the sequence? So the specific instances I was showing for the in situ sequencing, we can do up to 60 cycles of next-gen sequencing on these fixed tissues. So we're talking on the order of 60 base pair long sequence tags. The reason we can see single base differences because we're in the same way you can see single base differences in conventional sequencing, but this is conventional next generation sequencing. You're adding a fluorescent base or ligating on a base specific short hexamer oligonucleotide, and this gives a very crisp single base discrimination, and so that's how we do it. Another question here from Robert Noble. From a more philosophical viewpoint, how do you see the action of expression on a genetic basis? How does this impact our psychosocial components? I'm paraphrasing here slightly. Many of which happen in very short periods of time. Most of the methods we're talking about here are either snapshots of fixed tissue, but there are certainly some of the traits that we want to be able to explain cause and effect, where one's inherited genome can impact over long periods of time your day-to-day, even millisecond interaction with your environment by combining such real-time monitoring as EEG, EKG, fMRI that varies from person to person in combination with the environment, and seeing how that person-to-person variation is reflected in the genomes and epigenomes. Michael Furtaw from Lycor Biosciences asks, will genome sequencing be able to predict post-translational modification of proteins such as phosphorylation and glycosylation? I think what we're talking about here is not so much genome sequencing as sequencing, next generation sequencing, which can include nucleic acid tagged antibodies. So by some of the same methods that I mentioned earlier, where we can find what your own immune system is thinking as it's exposed to the environment, we can also use this for finding ever richer sets of antibodies that are specific for specific proteins, their post-translational modifications such as phosphorylation and glycosylation. Many of these have already been established through more classical polyclonal and monoclonal antibody searches, but I think this will ramp up very, very quickly with these new next-gen methodologies, and this will allow us allow us to detect in situ proteins the same way we're beginning to detect in situ RNAs. It will probably be initially protein subsets. Now on the order of thousands of proteins that are available, you might even want to take subsets of those, but I think that's a very interesting line of exploration. Okay. Please keep your questions coming in. Here's one about what are the prospects and status for interpretation of personal genome data. I think this is one of the key things that we need education and communication about how well interpretation does work. It's very easy to look at the half-empty part of the glass, that there are many things we can't do with either genome sequencing or other kinds of genomics and omics, but there's so many things we can do, and this is true of almost every medical diagnostic practice. There's a subset of applications that work, and so with the inherited genome, the half-full part of the glass is that we have 2,400 diseases which are highly predictive and medically actionable, and these need no longer be restricted to people who have had family examples of these genetic diseases, because there are many examples where you'll be the first person in your family because of the presence of a recessive allele from both your mother and your father. You're the first person to have it, and so you can't exclude those, and since you can't exclude any particular gene, that means basically all of us are at risk for every genetic disease which is and those that are diagnosable right now, the 2,400 that are highly usable, could be used pre-conception or premaritally as you're making reproductive choices. There's a question here about how long until the newest technologies are available, and will any of the current technologies be displaced? I think we have a history already in the very short time that we've had next generation sequencing of the various companies merging and producing hybrid methodologies. And there inevitably will be some displacement. I think that the two versions that are coming along quickly now are this in situ sequencing, which will have a whole new set of applications not currently captured by by current methodology. And then, of course, the nanopore could open up new markets, not necessarily displacing anybody because of the high portability or the very rapid response to potential environmental pathogens, possibly as little as a few minute delay between the incoming particles and the outgoing sequence interpretation. Let's see. Okay. Yeah, why don't you read it, because I need to be logging in here. Okay. Right. So, can you use fresh tissue samples with FISSEQ? This is from Herb Thoreau from Helix to Health. Yes, you can use fresh tissue. It's fresh at the time you start using it. It is fixed at the time that you're doing the sequencing itself. Yeah. So it's initially fresh, but so you're certainly not limited to cell lines or to to old pathological samples. Fresh tissue is quite usable. Here's a question from Arling Hing Lo. Regarding the use of HIV antibody CDR3 for therapy, would the 3-year period of immunogenetic detection supersede vaccine production? Let me reframe this slightly. So the antibody therapy is still just in rodent testing. It looks quite promising. In principle, it should be usable as long as the muscle cells keep producing the antibodies. We don't know how long that will be. It could be the lifetime of the individual. It could actually be longer than conventional immune cells, in that the muscle cells turn over more slowly, say, than some of the memory B cells. I think that's speculative. Time will tell, but it could be better than conventional vaccines. How many you can have operating at once, and what kind of load you can handle in your muscle and in your blood is also an open question. Right. So here's a question from Robert Noble. Is the PGP only for human genomes, or will include all genomes from the local biome for immune cross-referencing? Certainly, we already have examples of multiple microbiomic components and we're beginning to get the immune cross-reaction panel up, and it's certainly our ambition as a community, and this can be broadly shared like Wikipedia, to get a diverse set of genomes, including plants and animals that constitute our food and our environment. Yeah. Right. So Parvin Metaphore from Tehran asks, how do you assay telomeres with FISSEQ? This is we're haven't done telomeres with FISSEQ yet, but it would be very similar to the way that we would analyze telomeres in any next generation sequencing, which currently next generation sequencing has trouble with long tandem repeats. And so one of the things we hope FISSEQ will do is by having stretched long repeats, we can handle a variety of new projects. Here's a question of how can we participate in the Personal Genome Project. You can go to personalgenomes.org, learn more about it, and eventually take an entrance exam. Darlene Bunton, University Health Systems, says that enjoyed the presentation. Joseph Abbey from Venture Group says next generation sequencing and other related genomic technologies. How global is the effort to generate a more accurate primary data for diagnostic and academic research? This is a very interesting and timely question. The U.S. National Institute of Standards and Technology, or NIST, is currently generating a standard that will be widely shared. One of the ones under consideration, and this is a transparent conversation that's going on publicly, go to the NIST site to get more information, called Genome in a Bottle, and they are that one of the sample types that's under considerations are trios, possibly as 8 trios initially from the Personal Genome Project, which will allow us to generate more and more accurate primary data. I mentioned 1 in 10 to the 7th, meaning 1 in 10 million is sort of where we are. It would be nice to get closer to 1 in a billion, or less than 1 per genome, 1 error per genome. Thank you all for your questions and comments, and I would encourage you to continue to participate in this wonderful community that we have here, and attend to other BioConference Live. The personal genomics and do-it-yourself biology community involves researchers around the world, and the sharing that you can do is highly encouraged. Thank you again, and have a good day.